The thing that makes DNA very unique is that it's the only substance capable of replicating itself. During S phase of interphase, DNA doubles. It takes a few hours to copy the approximately 3 billion nucleotide pairs. Now that's pretty fast. If the copy rate was 1 base pair a second, the process would take 100 years to complete, and the accuracy is near perfect, about 3 errors in every round of DNA replication. Each strand of DNA serves as a template for the creation of its complementary strand. It's referred to as semi-conservative replication, where each new molecule of DNA contains one strand of the original molecule and one new complement. The new DNA molecule conserves half of the original molecule. During S phase of interphase, DNA undergoes semi-conservative replication. Each new molecule of DNA contains one strand of the original and one complementary new strand. Replication begins at the replication origin. There can be thousands of replication origins along the length of DNA. During interphase of cell cycle, DNA shows as chromatin. Each strand of DNA will supercoil into a chromosome during prophase. So for now, during S phase of interphase, imagine the DNA as uncoiled chromosomes, or chromatin. Helicases bind to the origin and unravel a segment of the helix. Helicases represent a group of enzymes that unwind the helix and opens up the two strands by breaking apart the hydrogen bonds between the nucleotide base pairs. This forms a replication bubble. Free-floating nucleotides found in the nucleus bind to the now exposed nucleotide bases. If a thymine is exposed, a free-floating adenine attaches by way of the DNA polymerase enzyme. Neighboring exposed nucleotides along the original strands are paired with their complementary free-floating bases, expanding the replication bubble in both directions, a process called elongation. Odd as it seems, and a little beyond the scope of this course, is the way in which the nucleotides attach. Since the two original strands run in opposite directions to each other, anti-parallel, remember, it turns out that the nucleotides are added to the parent strand in one direction only. That is, the upper blue strand, say, sees the pairing of new nucleotides in a right-to-left direction, and this is called the leading strand, while the lower blue parental strand sees the pairing of the new nucleotides in a left-to-right direction and is called a lagging strand. The developing lagging strand is made up of Okazaki fragments, joined to form a continuous strand by DNA ligase, like a molecular glue. Another job of the polymerase enzyme is to proofread the connecting nucleotide bases to make sure they're correctly complementary. If not, the offending nucleotide is cut out and another is ligased back in. Nucleotides already in the nucleus attach to the exposed ends of the parent strands. Nucleotides are derived from our diets, from the cellular material we eat found in plants and animals. DNA polymerase connects a nucleotide to its complement on the parent strand. DNA polymerase also acts as a proofreader to ensure the nucleotide being added is the correct complement. Elongation extends the replication bubble by the addition of complementary nucleotides at the replication fork. Replication terminates when there ends up being two identical strands where there was once one. DNA sequencing. Identification of the precise sequence of nucleotides along a DNA fragment. Different to gene mapping, where whole sections of nucleotides form a gene, a mapping of the gene tries to identify where genes are along the length of a chromosome, DNA sequencing identifies all the nucleotides along the length of the entire chromosome. The Human Genome Project sequenced the entire human genome. Expected to take many years, the Human Genome Project, started in 1990 by James Watson, yep, the guy who started it all, was finished way ahead of schedule in 2003, as new techniques were developed along the way to speed up the process. The publicly funded project was handed out to science institutions and universities, but a private corporation, Celera Genomics, used a new technique that enabled the rapid sequencing of huge amounts of DNA at a time. The intention was to secure the rights to privately owned genes, and if they were found before the Human Genome Project found them, then there would be gaps in the public display of the human genome. President Clinton announced that the genome sequence could not be patented and made it freely available to all researchers. This presidential announcement caused Celera's stock to fall hard, and in 2 days the biotechnology sector lost $50 billion in market capitalization. The genome is the sum of an organism's DNA. The human genome has about 3 billion nucleotide bases in a haploid cell, double that in a diploid cell. The number of bases does not bear any relation to the complexity of an organism. A single-cell protozoan species has over 650 billion bases. Genes are the functional subunits of DNA. Scattered among the 3 billion bases are the genes, about 24,000 genes. Again, the number of genes fails to correlate to the complexity of an organism. A species of roundworm has as many genes as a human. The irregular spacing of the genes means the chromosome size is not necessarily related to number of genes contained within it. The large chromosome number 4 has only 800 genes, while the smaller chromosome 19 has 1,500 genes. It turns out that most of the DNA is not connected to gene function. This so-called junk DNA has nucleotide sequences specific to the individual, and it's these sequences that are looked at in DNA analyses during crime scene investigations. RNA. The nucleic acid RNA has these differences between it and DNA. RNA is a nucleic acid and is important in the transmission of genetic information on the DNA to the rest of the body. DNA, for instance, is found in the nucleus, but RNA is found in both the nucleus and the cytoplasm. Although arguably DNA can also be found in the mitochondria, and in plants it can be found in the chloroplast. DNA contains thymine that binds to adenine, whereas RNA doesn't contain thymine at all, but it does contain uracil, which also binds to adenine. DNA is a double-stranded helix, where RNA is a single strand and non-helical. And the physical difference between DNA and RNA is that DNA contains a deoxyribose sugar, whereas the sugar molecule is just simply ribose sugar.